And the next speaker is going to be Brandon Benson, who is up here on screen already. And Brandon is a PhD student at Stanford, working in the lab of Dr. Ganguly. And today he is going to present his most recent research on critical behavior networks of inhibitory and excitatory spiking neurons with weak synaptic efficacy. So Brandon, go ahead. I don't
Thank
see
you.
your video, but yeah.
Can you hear me and
Yes.
see my slides?
Yes.
Great. Yeah, so I'd like to start with this recent collaboration that I was a part of with the Ganguly lab and Diceroth lab. It was published last year. Particularly highlight this computational collaboration. James Marshall, Timothy Mikado, and Sean Quirin here did the experiments and the analysis. And then myself and Jonathan Cadman did the theoretical work and simulations. So in this experimental work, a mouse is trained on a go, no-go task where the mouse is shown a vertical grading. When it's shown a vertical grading, it learns to perform a behavior, in this case, licking. When shown a horizontal grading, it does nothing. The neural activity is recorded during this task. We are able to identify subnetworks of cells that prefer vertical grading and horizontal grading. Once these subnetworks are identified, the visual grading is slowly removed. Eventually, the optogenetic stimulus alone can elicit the behavior. And that's the main point here, that stimulating the right subnetwork of cells causes the mouse to do the behavior. This is successful even without the visual grading. Even more surprising is that a small number of stimulated cells is enough to elicit the correct behavior. So we estimate that this subnetwork of cells contains at least 5,000 neurons, but stimulating only 20 or less neurons is enough to cause the correct behavior. How is this possible for the system to be so sensitive? When we look at this subnetwork, here 20 neurons is enough to recruit activity of a substantial fraction of the whole network. So again, we're seeing a small number of neurons that causes a response from thousands of neurons. The main takeaway from the experiment is this puzzle. How can only 20 neurons be enough to cause the response of the whole network? To answer this question, we start with the back of the envelope calculation. We set up a Poisson model using biologically plausible parameters. We set a threshold. If mean firing goes above threshold, then a large event takes place. And if it's below threshold, the system is stable. So we find that we can choose, with a biologically plausible set of parameters, we can choose the threshold so that a false event from the noise happens very rarely, only every 19 hours. But a small stimulus of only 30 neurons is enough to push the firing rate over threshold and cause an event. So this is a nice back of the envelope calculation. It could be a lower bound, but can we actually construct a model, a simulation, where 30 neurons is enough to cause a thousands of neuron size event? So we'll start with a rate network. I'm gonna set up this population of neurons with firing rate S, which is determined by the sigmoid of the membrane voltage and a threshold. Membrane voltage evolves according to a leak term, recurrent inputs, and a noise term, where the recurrent inputs are given from the excitatory population and inhibitory population from an external population, as shown here. So now we might have thousands of neurons in the system. We want to make it easier to analyze. So we'll apply mean field theory. So instead of looking at the individual firing rates, we look at the mean firing rate of the whole system. So we define this mean field M, and then we define the mean field of the inputs. And we're able to solve for self-consistent relations for the stationary solution using mean field theory. So for example, here we have an equation for M as a function of U, and for U as a function of M. And H here is just the margin of a Gaussian. So we've solved for the stationary states of the system. But we can learn even more about the dynamics. We can, for example, consider this phase space of the mean activity of the excitatory population and the mean activity of the inhibitory population. For fixed excitatory activity, we can solve for inhibition. And we call this the inhibitory null line, plotted in orange. The system will flow towards this null line. We do the same for the excitatory null line. The system flows towards this, so the solid blue line, and the dotted blue line is unstable. So when the two solid lines cross, the system is at a fixed point. So now we can, when the system's prepared at this fixed point, or first we can tune the weights of the network so that this fixed point is very close to the unstable line. So what does that mean? If we give a small stimulus and perturb the system, we might get some modest dynamics that look like this trajectory. But one trick we can do, and again, what we're looking for is a very small stimulus that causes a large event, not just a small one. So if we make the inhibition slow, then relatively we move much faster in the right and left directions on this plot and slow up and down. So now a small stimulus will cause us to flow very quickly towards the stable point here, and then slowly up to the orange null line. And when we're off this null line, then the stable point's back down here, so we flow back here. Am I back? I just got a notification.
Yes, you're back.
Okay, thank you. And so slow inhibition is this trick we can use to get the phenomena we want. This small perturbation creates this large response where almost the entire network, the mean of the network, has jumped by 1. Okay, so we run this simulation of this network, and indeed, stimulating 30 neurons in this window is enough to cause this order n large response increase in the mean firing of the network. We don't have to do it just for 30 neurons. We could do it from 0 to 50. We see this nonlinear response that 30 or more neurons is enough to reliably cause this phenomenon. So we used a rate model for this, and that assumes asynchronous spike times. But we tuned the network to this critical point where it's very close to instability, and it's unclear whether our asynchronous assumption holds here. So we might look at a synchronous network, like a leaky iterate-and-fire spiking network. And we follow the pattern of Brunel in his work from 2000. And that's cited right at the bottom here. So we'll have a membrane voltage that evolves for each neuron. There's a leak term, recurrent input spikes, and external spikes. So as the voltage integrates, it integrates towards a threshold, just 20 millivolts in our case. The neuron then fires a spike, and it resets to 0, and it stays there for a refractory period of 2 milliseconds. Meanwhile, the spike has a transmission delay of 1.5 milliseconds. And then we choose the connectivity as so. We have a graph here chosen connectivity 0 or 1, and then the weights are weak. So we have J normalized by the average in degree of the average excitatory in degree. And the external input we approximate as this constant term plus noise. And again, we can tune the right parameters of this system. We find that in a certain region, stimulating 40 neurons at saturated firing rate is enough to cause this large event. So this is an order n response. If I sum up the number of neurons that spiked during this event, I get about 0.97. But something's different here. We haven't added slow inhibition like we did with the rate network. So how can this network undergo such a large response and still be stable? So we can dig into this model a bit more by considering the evolution of the membrane voltage. At a specific point in time, I can plot the distribution of membrane voltages across the network. So this is the stationary state before I perturb the system. You can see most of the voltages are near threshold, close to firing. I do this again as the network excites, and I see that about half of the neurons have reset back to 0. After the event occurs, most of the neurons, or all of the neurons, are below threshold, have reset. And then this bump evolves back up slowly towards the stationary solution. So the key thing to consider here, why is the system stable? Well, at this point, why are all of the neurons much below threshold? All of the neurons have fired. This is an excitatory event. Why are they so low?
1 minute.
So with a back of the envelope calculation, again, we have a full mean field description of this, but again, back of the envelope, the mean input for a large event is going to be J1 minus gamma G. And that's an excitatory event, so that the size of that is about this. But the threshold causes each neuron to reset from 20 back to 0. So there's an effective inhibition here, and there's a battle between these two. And the 1 minus gamma G is always between 0 and 1. So if I drop that, then we have this reset stabilized region, where the reset's important. And in this region, we can look around the phase space. We see rich activity, synchronous activity, oscillations, stationary states. And in the future, we want to dig into this more. How does it relate to critical excitability and to what we observe in V1? Can we find phase transitions in this space, in this limit? And how does it relate to recent work of oscillatory models and critical avalanches? So thank you for listening.
Thank you very much, Brandon. It was really great talk. Again, I'm gonna clap for everyone here, and while waiting for the Q&A, while waiting for people to ask questions, I have a question myself, which is,
Sure.
so in your first model, you were taking like this 30 neurons as a number of neurons to activate, right?
Yeah.
Simulate, and then at some point you move to 40 in your second model. How did you make out, like, kind of figure out these numbers? Like, why 30 neurons and not 25 or 50?
So it's a little arbitrary. I could have chosen to show in the figure anything between 0 and 50 in this case. In the later model where I showed 40, that's because it was a larger network. Mm-hmm. So you tend to need slightly more neurons to elicit the behavior.
So it's a sort of a proportion of the size of the network, like is a sort of proportion to the size of the network. The number
it grows with some function of the network, yeah. But,
okay.
Yeah. But we want to keep the number very small.
Yeah.
Close to 1.
I see. Okay. As there are no questions yet coming, yeah, I want to ask a further question here, and is like, how fast are the simulations? So in the previous talk, we talked briefly about deep learning and like GPU sort of processing. Like, how fast are these models to be estimated?
So these ones, for example, I ran for 600 milliseconds, and I'm just showing you the final 100 milliseconds. Mm-hmm. And they ran in Julia in a couple minutes.
Okay.
So, but in Python, they're much slower.
Yeah.
I even tried to GPU, but Julia is just super fast.
Yeah, definitely. All right. So yeah, let's thank Brandon once again. And if people have questions, like, feel free to use the chat to ask them. But in the meantime, we can have our next speaker.